Good afternoon, one and all. Am I audible? So, welcome back, everyone. It's great to you have all back after the short break. I am Ambar Shruti. Once again, welcome you all on the behalf of Dayanand Education Society, Dayanand College of Pharmacy, and Dayanand Institute of Pharmacy for this one-day national level webinar on advances in the pharmaceutical sciences, the synergy of artificial intelligence, machine learning, natural biology, and regenerative medicines. Now, we have completed our two technical session at morning. We proceed with our next session of webinar with respected guest speakers. Now, heartfelt welcome to our next speaker, respected Professor Dr. Said Shadab Raza sir, Lab Stem Cell and Restorative Neurology, Department of Biotechnology. Also, a warm and heartful welcome to respected Dr. Uday Bhosle sir, Head of R&D Lifestyle Retail Private Limited, Hyderabad. Now, we proceed with the next of session. I would like to invite Mr. Kondapure sir to introduce our next guest speaker. Over to you, sir.
Thank you, ma'am. Very good afternoon, one and all present here. This is Arun Kondapure, Assistant Professor, Dayanand Institute of Pharmacy, Latur. It gives me immense pleasure and honor to introduce our guest speaker for session 3, Professor Dr. Said Shadab Raza sir, a passionate academician and researcher. Dr. Raza is a professor of biotechnology at IRA's Lucknow Medical College and Hospital, IRA University, Lucknow. He also serves as the in charge of Department of Stem Cell Biology and Regenerative Medicine at IRA University. He is also a visiting faculty member at the American University of Barbados in Latin America. Dr. Raza did his PhD in toxicology from Jamia Hamdard University, New Delhi in 2012. Subsequently, he became a postdoctoral fellow at the Cleveland Clinic, USA on stem cell and nanomedicine therapy for neurodegenerative disorders, followed by another postdoctoral fellow at the University of Utah, USA on stem cell therapy for multiple sclerosis. He then started his lab at IRA's Lucknow Medical College and Hospital in June 2015 as a group leader. As an internationally recognized scientist, his research excellence is reflected in his research achievements, internationally impactful publications, several recognized awards, the acquisition of third-party funds, and invitations to various internationally reputed meetings as a speaker. He also serves as an editor and reviewer for several leading journals. In pursuit of the best academic opportunities, his education, training, and independent research has spanned seven countries in the last 2 decades: USA, Germany, Australia, Japan, Italy, the Czech Republic, and India. Dr. Raza's impressive discoveries have made significant contribution to basic biology and disease mechanisms. He is the inventor of 3 technologies. The first one, the spinal needle hook technique for inducing ischemic reperfusion injury. The endothelial one induced permanent ischemic technique in a 3-day-old chick embryo, and asphyxia apparatus to induce hypoxia in Drosophila melanogaster. In line with his groundbreaking research, he has published over 70 articles in prestigious journals such as Stem Cell Reviews and Reports, Stem Cell Research and Therapy, ACS Nano, Chemical Engineering Journal, Brain Behavior and Immunity, Neuroscience, and Journal of Visualized Experiments. Among his 70 publications, he has achieved a remarkable total impact factor of 464.4 with an average impact factor of 6.69 and the citation count above 3,000. Dr. Raza's work has been recognized with several national and international awards and honors, including the European Molecular Biology Organization Travel Award 2023, the Indian National Science Academy Visiting Scientist Fellowship 2022, elected as a member of Leadership Development Program, Asia Regional Early Stage Professional Subcommittee, Fellowship 2011, IBRO, that is International Brain Research Organization for Italian Neuroscience Young Investigator Award 2011, IVRO Travel Grant 2008. So we are fortunate that such a speaker is with us today. Today he is going to speak on the topic stem cells and biomaterials, a new era for ischemic stroke treatment. Now I feel pleasure to invite Professor Dr. Saeed Shadab Raza sir to kindly share his insights on the topic with us.
Should I
Over to
start,
you.
Dr. Arun?
Yes, sir. Yes.
I think there is some slag in the connection. Can you hear me?
Okay.
Hello?
Yes, yes, sir. Yes, sir.
Okay.
Yes.
Let me share my slide. So first and foremost, very good afternoon to all of you, and I'm so thankful to Dr. Arun and also Dr. Wajid for giving me this opportunity to deliver a talk today at your prestigious institution, Dayanand Institute of Pharmacy. So my area of research is stem cell therapy for neurodegenerative disorder. Specifically, we are interested in ischemic stroke. So today I'm going to deal with stem cells and biomaterial, which is a new ray of hope for the treatment of ischemic stroke and related disorders. So to quickly start with, as I just mentioned, that in our lab, we mimic ischemic stroke in rodents, Alzheimer's disease. We study Alzheimer's disease both in rodent as well as in fly. We also study ischemia reperfusion, other sort of ischemia reperfusion mechanisms such as hind limb ischemia, such as ischemia reperfusion in embryo that I'm going to show at a later stage in today's talk. Beside this, as a therapeutic, we use primarily we use stem cells, followed by some nanomaterials, nanoformulations, or some novel molecules like currently we are using gas releasing peptides. Beside that, the mechanistic approach that we utilize is NLRP3 in inflammasome studies and autophagy since the inception of the lab. By the way, I would like to, I would love to let you know that today I'm completing my 10 years back in India after my PhD and a postdoc in United States. So, and also I was there at the very first session and the second talk by Dr. Professor Zinat Iqbal. And after that, especially the talk by Zinat Iqbal was awesome, and in such a depth she has intimated us about the AI-driven pharmacology treatment. So there is very little room for me to go beyond that. But since I am more into the research rather than I'm a teacher, I do a lot of research in my current institution. So I will make you acquainted with the results that we have received till date and that we have published, and some of them which are on the verge of publications. But before that, I would like to introduce the basic of ischemic stroke recovery. So as perhaps you all might be knowing, because as far as I know, Dr. Wajid has yesterday informed me that several B Pharma students and followed by M Pharma are there. So let me go to the very basic of the stroke and its type. So stroke is basically the impedance of blood flow to the brain. I believe you might be knowing, and it is broadly divided into two types. One is ischemic stroke, and the another one is hemorrhage. So hemorrhage is further divided into intracerebral and subarachnoid hemorrhage, but it consists only a very few population, that is approximately 12 to 15% of population gets hemorrhage. Rather, most of the patients that we see is because of the ischemia in some one or two arteries in the brain. So most common of them is middle cerebral artery. So in our lab, we mimic middle cerebral artery occlusion in the rats that I'm going to show you very soon. So majority of cases, that is 85% to 87% of the cases are of ischemic type, and that is why most of the lab across the globe mimics ischemic stroke rather than hemorrhagic stroke. So quickly coming to stem cells, since our primary interest as a therapeutic is in stem cells, so we transplant stem cells to the ischemic brain as well as also to, in some cases, to Alzheimer's brain too. So if you will ask about what is to me that what is a stem cell, so stem cell is a naive cells. Naive means that it is a blank cell or it is a mother cell from which you can get once it divide it produces specialized cells as well as again mother cells. So the beauty of this cell is that it has a unlimited capacity of renewal, so it keep proliferating. But the best part of the stem cell is that when it proliferates, one of its daughter progeny becomes exactly the replica of the mother cell, while the other one becomes a specialized cell. So this is the beauty of this cell. And as you perhaps you all be knowing it, that our body is the formation of our body is from these cells only, that I'm going to show you very soon in the next slide. So let us see on that on the basis of so okay, let me explain it in this way, that when a sperm and when a sperm and ovum fuses with each another, it forms zygote. So zygote is the first stage of formation of a whole human being. Okay, so at that stage, the cells that are present are often known as totipotent stem cells. So totipotent means that they can form only one type of cell. So once a zygote and ova is formed, fuse, it forms one cell. Then one will form two replica. Then two will form four replica of it. Four will eventually become eight. By this process, by the week 5 to 7, a blastocyst is formed. In the very next figure in the middle, you can see pluripotent stem cells. So the layer in the down is blastocyst. So that is pluripotent stem cell, and actually these are embryonic stem cells. So the source of embryonic stem cell is this blastocyst. We took stem cells from this blastocyst, and the formation of eventually embryo is from the blastocyst, and that is pluripotent. Pluripotent means unlike totipotent, which has a specific character of making only one subtype of cells, pluripotent can make any type of cell. So it can make your heart, it can make your kidney, it can make your brain, whatever you want is your wish, is can be derived from pluripotent stem cell, that is from the embryonic stem cells. So eventually when embryo grows to a fetus, so it becomes multipotent. This pluripotent stem cell, that is embryonic stem cells, it becomes more specialized, and that is what we call multipotent stem cell. So for instance, it can embryo can give birth to ectoderm cells, endoderm cells, or mesoderm cells. For instance, mesoderm is example of mesoderm is blood cells. So blood cells can be once it is specialized cells. For instance, it is a mesoderm cells. It becomes multipotent rather than pluripotent. What it says is that pluripotent can form every sort of cells, but multipotent can only form now because it is a mesodermal cell. For instance, it may be a ectodermal cells also, but if it is a mesodermal cell, it can only form blood cells. It can be form WBC, RBC, whatever it is. Okay, so only the blood cells it can make, and this is called multipotency of our stem cells. And then finally, once it is a multipotent, it will eventually form unipotent cells. So unipotent is like, for instance, if it becomes a WBC cells, it can now only begets a WBC, or otherwise it can only, if it is a neuronal, it can only produce neuronal. If stem cell is specialized for neuron, what we call neuronal stem cell, it can only produce neuron. It cannot produce glia or oligodendrocyte. So this property is called unipotent stem cells. I hope you might have a good understanding now about totipotent which is a single type of cell, pluripotent, which can give birth to any organ of your body, multipotent, which is basically a sort of adult cells. So either it can become a specific lineage is from this multipotent cell. So it can become either ectodermal cell or endodermal cell or mesodermal cell. And then when finally it is formed, then unipotent is only when you get somatic cells from stem cells. So that is a one type of cell. Only, for instance, only you are getting neuronal stems, and only neurons. Only you are getting WBC, the RBC. So now quickly going to the another stem cell side. Okay. By the way, I forget to let you know that in the previous this slide, we have defined the stem cell on the basis of their stages. So totipotent is a zygote stage, pluripotent is an embryonic stage, multipotent is a fetus, in between fetus and adult stage, and unipotent is of course a adult stage. Now on the basis of on the basis of differentiation. Sorry, I'm sorry. The last one was on the basis of differentiation, and on the basis of the stages, either the stem cell could be embryonic stem cells, so it can form only embryos, or either it could be a adult stem cells. So now coming to the main topic of my research. So in my laboratory, since last 10 years, we are looking into the fate of stem cells. What happens to a stem cells when we transplant it into a in vivo system? So actually, the fact is that if you will go to NIH site or any other clinical trials registry site, you will find that of course none. I mean, almost till date, 77 clinical trials have been taken up in United States only. So out of this 77 clinical trials in United States only, none has shown efficacy in terms of once it is transplanted, it is giving benefit to the patient. Whatever these many studies at different centers have shown that when you transplant the cells inside the in vivo system, it can only it would not be a harmful substance, but at the very same time, it is not giving benefit to the patients. So when I started my lab in India, back in India 10 years, major hurdle with stem cells is that when you transplant into the patient at a clinic, normally you do it through IV. So once you transplant a stem cell through IV, what happens is that rather than going to the target, for instance, in our cases, it is a brain, a stroke brain. So rather than going to brain, it follows the normal circulatory. So normal circulatory is that once you transplant it, it will go to the kidney, liver, lung, spleen, and then it will get excreted out. So the very first hurdle for a stem cell therapy for a patient is that target migration is lacking. The second important fact is that whatever cell is getting to your brain, for instance, in our case, only 1 to 3% cells survives after 28, almost 1 month of transplantation. So low survival and low target migration are the key challenges for a stem cell transplantation scientist. So in our lab, we are looking into how to make them the cells more home to the target for first thing, and whatever is coming to the target should stay for long and get embedded into the brain architecture. So coming to the next slide, this is what I said. Our lab investigate the fate of stem cell under stress. Okay, so once we transplant it in a Alzheimer brain or a stroke brain, then what is the fate of these cells? That is what we are looking for the last 10 years. So coming to these issues, the very first challenge for us is dose optimization. This is very important, that how much quantity you have to give to a patient. See, what is happening in a lab setup is that you transplant the cell in a controlled setup environment, so and you always monitor the your subject But unlike the clinical setups where you do not know what patient is eating, what is his habitual habit, where he is going, where how it interacts with the environment. So the very first challenge for us is that how much cell we have to transplant that can that should be enough to grow, to get embedded in the brain and get and get give benefit to the system. So that is the first thing. The second thing is that what should be the best timing after a stroke. That for instance, in normally what happens is that we have only one therapy against stroke, and that is TPA, tissue plasminogen activator. But the problem with tissue plasminogen activator is that you have to give it to the patient within 4.5 hours of window timing. Otherwise, the artery will become so weak that it will, rather than bursting the clot, it will burst the artery. So that is the major issue, and that is, unfortunately, we have only one treatment for ischemic stroke. The second being the surgery, which is not, of course, a feasible option for a country like India, for instance. Even in the United States, that is not a possibility. Who is in remote, he will come within 4 or 5 hours, and so that is a challenge. So the timing is a very important challenge for us. The cell number is very important. How much dose we have to give? I mean, whether we have to give just one time, we have to give two times, 15 times, 30 times. This is what we often standardize in our lab for different studies. By the way, in our lab, we utilize majority we utilize dental pulp stem cell, which is which is located at the third molar of your teeth, and followed by mesenchymal stem cell, which is a mesodermal, which has a mesodermal origin, and also neuronal stem cells. So these three stem cells we often use, but our prime choice is dental pulp stem cells. Now, please come to this figure. As I have informed you just now, that the problem with the stem cell is the very first problem is that target migration is lacking. So see, in this animal, after 24 hours, the of infusion of stem cells, you can see this stem cells can be found in spleen, liver, and lung, rather than going to the brain. So and after the next day when it was imaged, this paper was published in Stem Cell in 2022. So when next day it was imaged, there was nothing. I mean, almost everything was clear. You can go to the PubMed and search out for this paper. So the major hurdle is how to make them home to the target. So once it will reach the target, then only we can we can think of integrating stem cells into the tissue. So along this line, what we have done in the past, this is from one of my paper from my postdoc time. So we initially we decided whether we have to give the cells through IA or IV, that is intraarterial or intraventricle. So as you can see, this is a luciferase image assay that we have done in the United States. So as you can see that at day 0, when we transplanted through IA, that is intra-arterial, from artery rather than vein. So cells were, majority of cells were in the brain. So you can see the luciferase signal, which is coming in green from the brain. And in the figure number B, with days it was getting down, okay, in the similar 4 animals, but still there was a good amount of reflection of luciferase from these animals. But when we transplanted the cells in the IV, what we found that these cells were located in the abdomen, that is liver, kidney, and spleen. So that and by the day 7, it was all washed out. So exactly the same what I have shown in the previous paper by our colleague from stem cells, who has published in stem cells. The basic hurdle was to choose the best route, but the problem is with the human patient. In rodent, you can often expose artery and you can transplant it, but in human patient, normally the normal route of infusion of anything, even a drug, is IV. So that is a basic challenge that we have identified, that the route of administration of drug or whatever, or stem cell, is very important and very challenging. So this was the first key issue. The second one, okay, along these lines, currently we are working with Walzak Quater, one of a very good friend and colleague from University of Baltimore. So what they are developing is that this is called stem cell image guided therapy. So image what they do is that they transplant a stem cell through IV or through IA in patient under the guidance of MRI. So for this, what we have to do is that what they are doing, they have already published it, and still they are working on it. We are also collaborating with these people. So what you have to do is that you have to make a stem cell, labeled with iron oxide or something, spion or quantum dots, and then you can, under the influence of, as you can see, this is PET MRI. So under the influence of the magnetic field of PET MRI, those you can attract those stem cells because they are labeled with spion or iron oxide, so they get attracted. So in the figure number F, you can see on the right atrium where there was, or also in the 3D image of H, you can see on the ipsilateral hemisphere, those that is ipsilateral means the area where the stroke is really happened. Okay, so so you can see the majority of cells were attracted to the ipsilateral hemisphere. So this is a new technology that is widely going on in United States, and we are a medical college, so we are collaborating with these people, and we are looking forward for in next 2 or 3 years to start this particular technique here also. But of course in rodent, not in human. So currently with these people and with another friend, Johannes Boltz at Warwick, since we do not have permission to utilize PET MRI for what currently in our lab we are doing, we are making them making the cells lentiviral positive for chemotactic factors. So chemotactic factors, if you do not know, are those factors which attract cells from distance. Okay, for instance, if you have an injury in any sort of injury, that hence leading to a clearance of the injured site. So exactly utilizing the same technique, what we are doing at the moment, we are utilizing ICAM. ICAM is adhesion, intracellular adhesion molecule. We are attaching this intracellular adhesion molecule with VEGF on the surface of the capacity. The VEGF is and ICAM, CHCR, these all are known for. This is what we are currently utilizing, but our main aim is to utilize ICAM. So these are basically a chemo attractant molecule. So along the direction of the injury, they automatically move. So what we are doing, that we are attaching these molecules on the stem cell surface. How we are doing? Let me show you in the next slide. So we are utilizing lentivirus preparation method. So there are broadly two methods through which you can either inject some cell with those proteins. So ablate means you can remove the protein or you can add the protein. So for instance, we are adding ICAM. So what utilizing lentivirus. So there are two. Transfection is a very common process in molecular biology. But this utilizing lentivirus will bring a permanent transfection. So what it means is that because lentivirus is a virus, so utilizing the genome of this virus can grow, because it is a virus, it can grow indefinitely. So that is why we use lentivirus. And we if you will see on the very left, transfer plasmid, down of the transfer plasmid, what we do, we combine transfer plasmid, packaging plasmid, and envelope plasmid. And the packaging plasmid has our cell of interest. So if we want to, for instance, ORFs are present, ORFs are genes are commercially available in the market. So ICAM genes are commercially available in the market. You can grow, you can purchase them, you can grow in with E. coli in the lab. That what we do in the lab. And then finally, when you purify those plasmids, you can transfer these two with the genes of lentivirus. So what happens is that finally, when they combine with each another, they start producing a unlimited, expression of that particular gene. For instance, in this picture in the middle, you can see a green color. Our gene has our gene was tagged with a GFP. And so this is why you have to do this process. So for instance, if you have to transplant the stem cells, you will never come to know in a patient or even in a rodent, if it is alive, you will never come to know whether this the cell which you are examining is from the inside of the body or whatever you have transplanted it. So to monitor the cells which has been transplanted in the system, you need to have a color which can distinguish it from the normal environment of the body. So that is why we do it. Okay. And since it has a lentivirus, so it can replicate again and again and again. So you can always monitor your stem cells where they are going on. Okay. For instance, in this case, in our lab, we are using GFP. Okay. So you can see this figure, HK2. In this one, what the strategy is to purify it first. You need to have these three complexes together in a HK293 T cells. This is also a virus cell, SV40 cells. And then when there will be enough of this green color, they will start excreting out this plasmid. This green color is nothing else than plasmid. So once it is excreted out, you can you can take them out. You can purify them with the puromycin selection, and then you can you can know what is the MOI of this. Is what we normally we do. If you see this green box in the center, we will see on the very first day the MOI was just 0.29, but on eventually as we started playing with the composition, it becomes 2.08, 235, 235. So you have to determine which is the best MOI for your study. Okay. This is what we do. And then we isolate finally. Okay, then we isolate the plasmid, and in the down figure, if you will see, this is more green. So these are the down ones are stem cells, the upper one are HK293. HK293 you have to utilize for making the virus, and once the virus is made, it is purified, then you have to transfect it into the stem cells. So these are all stem cells. Some are with high color and some are with low color. So finally, you have to either do a pure I mean, puromycin selection or whatever newer plasmid has a sorter, and then you can sort out the green color, whatever intensity you want, and you have a purified population of stem cells. So once these stem cells are purified from this is all what we do in our lab always. Okay, then only you transplant it into a rat or into a mouse. We do it into a stroke driven rat. Okay, so now we have once we transplant it after 28 days, 6 months, whatever the study time period is, then we normally what we do is that we examine once we sacrifice the animal, we slice the brain, and then we examine it under a normal fluorescent microscope. Whatever color, if any cell is giving a color, that says that this is a stem cell which is embedded in the brain. Okay, I believe I have done justice to let you know what is how we are doing this lentivirus transfection. Okay, So anyway, now coming to middle cerebral artery occlusion, this is MCO is middle cerebral artery occlusion. As I said, ischemic stroke is the most common type of stroke, and it often happens because of the occlusion in middle cerebral artery. So you have to mimic middle cerebral artery occlusion in your rodent model also. This is what we do, and normally currently we are also trying on the very right side. If you will see A and B, we are trying to transplant stem cell through common carotid artery. So if you will put your two fingers on the either on the right or on the left side of your neck, you will find a pulsation. This is your common carotid artery. This is right common carotid artery, and this is left common carotid artery. So CCA is a left common carotid artery, and we are transplanting stem cell directly into the ICA. ICA, it further common carotid artery further goes inside and get get splitted into ICA, that is internal carotid artery, external ECA, external carotid artery, and pterygopalatine arteries. Okay, so we are directly trying to transplant stem cells into the ICA. Beside and examining it with the transplantation through IV. Okay. Now, since I have already informed you that transplantation is homing is a key challenge. So for this, we have we have created our own model in a 3D developed chick embryo, perhaps the first ever model of ischemia reperfusion in an embryo. In between, that is embryo is growing, and on the right and the left side is the vitelline artery. Vitelline arteries are responsible for bringing food and oxygen to the embryo. So right one is right vitelline artery, and the left beneath the trunk, beside the trunk, and the left one is left vitelline artery. So on the figure number, if you can see on the what we have done is that we utilize a spinal needle, which is oftenly used for infusing drug in the spines of the patients. So that we utilize, we custom designed it, and we bent it the corner of that one to lift up the right vitelline artery. So in the figure number F, you will say we have occluded the right vitelline artery, so that it created the ischemia region on the right side, and with the aim to transplant transplant stem cell on the left vitelline artery. So I'm going to show you quickly a video of it. It has already been granted a patent, Indian patent. US patent has been submitted for it. We have published it in Jove and also in Pharmacologica. So I think you might have a better idea of this one with this video. Now, quickly coming to the Okay, in this picture, as I have said that on the right vitelline artery we occlude the artery, and on the left side we transplant stem cells. This girl is trying to inject stem cell through a stem cell injector. Hello.
Hello, you're audible, sir?
Yes. Yes.
Yes, sir, you are.
So this girl is trying to occlude to transplant the stem cells on the left vitelline artery. Okay. So okay, before I move on, please let me show you that MCO, which normally we do middle cerebral artery occlusion, and we leave animal for almost 20, a minimum of 21 days, compared to this hook eye. For this model, the model of chick embryo, we have given name of this model as hook IR. So for this model, you can see that whatever result we have got just in 6 hours, that is what we are getting in MCO, that is in animal in 21 days. So this model is very robust, very time effective, very cost effective, and currently not only we are utilizing it for stem cell studies, but also we are utilizing it for understanding the pathophysiology for ischemia reperfusion. Okay. Now coming to, okay, along this line, we have just published another paper in Bioprotocol 2025. This is a free paper to read. Please go and see, search it out in PubMed. So the previous model, if you recollect, we have occluded the right vitelline artery and then we have left it. So this is what we call a reversible phenomena. So I mean, occlusion is done for some time and then we reverse the process. But along this line, we have also created another model, that is a persistent ischemia reperfusion model. About this model, you can get more information in Bioprotocol, since we are running low of time. So I will skip this one. And then coming to stem cell survival, as I mentioned it very earlier, that our prime interest is not how stem cell is giving benefit to the system, rather what is the fate of a stem cell in once they are transplanted into the brain. So we are looking into stem cell survival death mechanism, and that is more specific to NLRP3 inflammasome and autophagy. So NLRP3 inflammasome, perhaps you people might be knowing, this is a nod-like receptor and is well known for DAMP, that is a damage associated molecular pattern. I mean, for diseases. This is in disease often it has been. What I want to say is that in diseases it has often been seen that NLRP3 inflammasome leads to inflammation of the system and leading to cell death. Okay, so what happened is that once NLRP sends a signal, danger signal, it activates ASC, that is picard protein, and picard forms an oligomer, and then finally it propagates its signal to caspase 1, which is in a immature form, and it eventually it cuts down and becomes a cleaved caspase. Once cleaved caspase is there, it becomes mature, then it cleaves pro interleukin and 18 and pro interleukin 1 beta into into cleaved interleukin 1 beta and cleaved 18. So this is what we are in depth. We are studying in many studies about the mechanism of how NLRP3 is associated with inflammation and how it is leading to the cell death post transplantation. So just quickly giving an overview of one of our study, which we have published in a stem cell reviews report in 2023, we found that OGD, that is oxygen glucose deprivation, this is a very common method to mimic ischemic stroke in vitro, in which what we do is that we deprive cells with oxygen as well as glucose both. What is happening to the cells? So we have found that OGD, that is oxygen glucose deprivation, promotes NLRP3 activation, which is in line with animal experimental stroke model. So if you will see on the figure A and C, the second row shows the OGD. So you can see the expression of these high level of proteins in Western blot, which was found found down-regulated with the use of MCC 950. So MCC 950 is another molecule which has been utilized by one of, which has been characterized and synthesized by one of our another very good collaborator from Australia. Her name is Avery Robertson. So we are also utilizing her formulations in our studies to examine the pathway NLRP3 as well as autophagy pathway. So coming to this figure number A, you can see that at OGD in figure number A as well as in figure number C, OGD is increasing the the concentration of these many NLR protein, that is not like proteins. NLRP3 inflammasome is a complex of, by the way, 7 proteins. Okay, so these all were get upregulated, but however with the utilization of MCC 950, which is a specific inhibitor of NLRP3, the expression was gone down, as you can see on figure A and figure C on the third row, as well as on the figure B and D on the second row. So the expression was gone, which was further correlated with SINLRP3 OGD. Am I on time, or should I cut down? Dr. Arun? Dr. Arun?
Yes, sir. You may proceed for 10 minutes.
Okay. So, okay, let's then skip it. Then if I have only 10 minutes, then we have also seen the expression of SIRT3, which is a mitochondrial protein, and it acetylates SOD2, and it is often regarded as a regulator of oxidative stress that we have also examined in these cell lines. We have also further we have examined the cell loss and cell survival utilizing the same mechanism of OGD and utilizing NLRP use, that is lipopolysaccharide, which is a well-known marker of NLRP3 inflammasome. So I will skip the results, but because I have too many of the results. So in brain, there are 2 regions which are neurogenic, unlike any part of the body. For instance, if you will cut out your tongue, it will regenerate. You will cut out some part of your liver, it will regenerate. Heart will regenerate. But unfortunately, the brain is the only organ which has a very limited capacity of regeneration, and it has only 2 regions where regeneration can be seen. One is DG, that is dentus gyrus. On the very right side, you can see the brown figure. DG is written in between. So DG can produce cells, and also SVZ on the left side. SVZ is subventricular zone. That is the only region where you find neuronal stem cells. Okay, so these are the 2 region. We have studied it in another study, in another stem cell figure in sub-figures. A C-shaped blue color dots are there. So those blues are DAPI, and this C is actually dentus gyrus. So this reflects the neurogenic region of the brain. Okay, so there are too many results related to this one. Anyway, now the next study that we do is of 35 proteins, and we are studying the mechanism of autophagy in our lab. So you can go and search out several papers. We have recently published stem cells. We have recently published SES nano and a lot of papers that reflects the autophagy mechanism. So I will not go into the depth of this one, but rather because I have too many. Okay, so I have also biomaterials. Okay, so we also utilize several nanoparticles scaffolds in our lab. So I believe that you must be knowing that biomaterials are the substance that can be used to integrate into biological system when used as a therapeutic or diagnostic. In this line, we have recently published, a couple of papers we have published, several papers, but this is one of the paper that we have utilized these silver loaded nanostructured lipids, NLCs, in human dental pulp stem cells as well as in mesenchymal stem cells. So silvestrate is an antioxidant molecule to we have, say, several formulation, characterization experiments, and then finally we characterize it on the different cells. Because it is a antioxidant, we utilize DHE, which is a superoxide marker, DCFDA, which is a pan marker of ROS, and also one X-ray, which is a H2O2 marker. So I will not go into these many results. These were too many results. Statistical analysis, we did one way. ANOVA Shapiro will test was confirmed through normal distribution. And coming to quickly to the conclusion. So the conclusion is that enhancing homing of stem and survival of stem cell is crucial for optimizing stem cell therapeutics. Newer models like what we have we are coming with is very important for stem cell homing, because stem cell is a very costly technique, so you must to have something which is very time effective, very less in terms of spending, it is very cheap, so and finally utilizing biomaterials alone or synergistically with stem cells can improve the transplantation, survival, and distribution of stem cells. So with this, these are our in last 2.2 year or 3 years, we have published these many papers as corresponding authors related to this work. And as far as the future studies are going, currently we are working with IIT Kanpur, and we are developing nano robots for stem cell targeted therapy with a group of Professor Sandeep Verma. And as I already informed, that for last 1.5 years we are exploring all the ways to work to get started on human NIH funded human project with Poiter on image guided therapy for stem cells. Currently, we are I have already shown. Currently, we are using CRISPR-Cas9 and lentivirus mediated stem cell delivery. So these people are our collaborator. Avril, as I said, MCC 950 was originally synthesized by her, and she initially published in Nature Medicine, and then later we utilized it for our ACS nano paper, blood-brain behavior paper. And I'm thankful to the funding agencies like UPCST, IBRO, and DST. This is my group. Majority of the work that I have shown was done by one of my former student, Ravi Prakash, who is currently in University of Michigan, and uh, and also now currently it is taken up by Arshi Vasi. So this is all I have for today. Thank you very much for listening so patiently. I hope I have done justice. Yes, sir.
Yeah, thank you so much, sir. Thank you
very much. I'm so sorry because I have too many of slides, and I didn't realize that unlike Zenith Madam, I will not get 15 minutes extra.
Okay. Thank you, sir.
Yes.
Thank you.
Thank you.
Thank you for such an informative and enlightening session. Your insights into stem cells and biomaterials, a new era for ischemic stroke treatment, have added immense value to today's webinar. Now we will move towards question and answer session. We have received a number of yes. So we have received a number of interesting questions from the students and participants and faculty members. Let's address one of them. Whether stem cells and biomaterials offer potential for treating diabetes and other metabolic disorders.
Certainly they offer potential for if they are giving synergistically, certainly, because the problem with stem cell, as I have already shown, I mean, this is true for every disease, not just for ischemia. Though ischemia is a very grave one, but still ischemic stroke. But this is true for every disease. We have gone through several clinical trials, and stem cells. I mean, stem cells by itself is not giving benefit to the patient because they do go somewhere else rather than going to the target, and because they only very low number goes to the target. So and because once they go to their target, for instance, in ischemic brain, So because of the hypoxic condition, because of ischemia, because of inflammation, they normally die. So with biomaterials, certainly we are, I mean, the at least clinical, the lab results are very wonderful for all of us if you will go to the PubMed, but clinically it is yet not proven.
Yes.
But we believe sometime, for instance, our own friend Poiter is working on a similar line,
Yes. Yes, sir. Sir, one more question is there.
Yes, sir,
How
please.
stem cells and biomaterial therapy is better than conventional methods for stroke treatment?
Sorry.
Yes, sir.
Oh, better
Yes, we
than.
have seen.
Yes.
Yes.
So see, for stroke treatment, there's only, as I already intimated you, only one FDA approved drug, that is TPA. FDA is Food and Drug Administration USA, and TPA is a tissue plasminogen activator. The problem with tissue plasminogen activator is that if it is not infused within 4 to 5 hours of ischemic stroke, the arteries will become so pale that once the drug is, what is the mode of action of this drug? Once we infuse this drug into the site of injury, in a ischemic region, so what is do is it bursts the clot, so it is a clot burster. So it bursts the clot. But what happens is that with time, after 5 hours, the clot will become so hard that rather than blustering the clot, it will burst the artery. So that is unfortunately we have only one drug that can be used. Okay, so you have to have go for another options, and the one very liable option seems to be stem cells, because cells are made of the one more important thing is that unlike embryonic stem cells, normally we transplant adult stem cells. The beauty of the adult stem cell is that when we take it from, even for instance, we are taking it from teeth, okay, which is a very feasible option. You can easily take out stem cell from your teeth. So once you take the stem cells from your teeth, you will transplant it into brain. It will start making cells of brain rather than making cells of teeth. So this is the beauty of adult stem cell. This is called co-transdifferentiation. So because of the virtue of transdifferentiation property, it will eventually start making cells of that particular area, whatever environment you are going to give it. Okay, so this is one of the feasible option. But unfortunately, in the last 3 or 4 decades, we have seen that there is no real outcome of stem cells. That is why currently we are utilizing scaffold and other hydrogels, etc., to infuse the stem cells into the patients. And this is giving at least benefit in terms of case reports, if not in a larger population. So we believe once it is sorted out, the low survival of and the low homing of stem cells, I believe it should be it will open a new window.
Thank you, sir. Sir, one more question is there. How close are we to seeing stem cell based treatment for stroke become a part of mainstream clinical practice?
No, honestly speaking, I will say at least not for next 2, 3, 4 decades. I mean, we are quite far, quite far. I will advocate this again and again. We are quite far, yes. So specifically in case of ischemic stroke, because it is a bedridden disease, it is, we are very far away, unfortunately. But still we are working, maybe one day, by that time I will be become old. Perhaps it will become a normal procedure.
Okay, thank you, sir.
Currently,
Thank you so much for
thank you
patiently
very much.
answering all the questions. It has truly been
Thank
a
you.
wonderful learning experience for all of us. On behalf of Dayanand College of Pharmacy and Dayanand Institute of Pharmacy, Latur, and all the participants, faculty members, I extend my sincere gratitude towards you for your time and expertise. With this, we now conclude session 3 of our national webinar. We will now move forward to the next session of the day. I would like to invite my fellow anchor, Mr. Kuldeep Pawar, sir, to take over and continue the proceedings. Over to you, Kuldeep, sir.
Okay.
Thank you.
Thank you.
Thank you. Sir.
Thank you, sir. Very good afternoon to all. I am Pawar Kuldeep, Assistant Professor at the Dayanand Institute of Pharmacy, Latur. It is my immense pleasure to be here with you all today, and I extend a warm welcome to each one of you. It is a great honor for me to introduce our esteemed guest for today's session, Dr. Uday V. Bhosle, a distinguished personality in the field of pharmaceutical research and development. Dr. Bhosle sir comes with over 15 years of experience in the pharmaceutical industry, particularly in the domains of Ayurvedic, herbal, cosmeceuticals, and novel drug delivery system. His core competences include product innovation and development, research leadership, regulatory and compliance, project and team management. Currently, Dr. Bhosle sir is leading the R&D function, that is research and development functions at Vidix and Skincraft, two pioneering brands in personalized skincare and wellness. Dr. Bhosle sir holds a bachelor's degree in pharmacy from BV Pune College of Pharmacy, Pune, MBA in marketing from Wadia Institute of Management, Pune, and master's in pharmaceutics and PhD in pharmaceutics from Rajiv Gandhi University of Health Science, Bangalore. His professional journey includes impactful roles at reputed organizations as Hindustan Unilever, Dabur International, Dubai, Wipro Consumer Care, and Ajanta Pharma, where he has consistently contributed to the innovation, formulation excellence, and strategic development. Dr. Bhosle sir has authored 19 research publications, delivered presentations at international forums, and has patented botanical formulations for anti-melanogenesis. He is widely recognized as a visionary research and development leader who seamlessly blends traditional Ayurvedic wisdom with modern scientific approach. We are truly privileged to have him with us today to share his valuable insights. Sir, we are honored to invite you to address the session. So, with the permission of Dr. Vijay Bhosle, sir, I hand over the session. Over to you, sir.
Yeah, thank you. Thank you, Professor Kuldeep. I hope you are able to hear me.
Yes, sir.
So,
You are audible.
yeah, this is the fourth session. So, actually, getting the fourth session and extending the fourth session will be a crime, actually. So, I'll not get this very deep, I could say, uh, either, I could say, but I'll make it interesting and I'll try to see that what we can take home from this session. Basically, I'll just share this slide first. Hello. Yeah. Can you see the screen?
Yes, sir. The screen is visible.
Okay. Okay. Okay.
So please on the presentation mode.
Yeah. 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 I'm doing that. Now you can see.
Yes, sir.
Yeah. So the today's topic basically it's AI application. So before starting, I'll just brief introduce about the organization where I'm currently working. So I'm working with Inknet Lifestyle. You may heard about the two brands, which is Vedix and Skincraft. Vedix and Skincraft. So it is India's first customized personal care product, basically. So when I say personalize or customize, means this product has been done, formulated as per the individual requirement, like that. And again, coming down to the basically Vedics, which is more or influenced by Ayurveda, and Skincraft is more it is influenced by Skincraft basically, or is more or influenced by synthetic. In my earlier assignment, I work with Unilever as a technical project lead. I work with Dabur, where it is basically for the mass market. So one fit for all solution. So this organization, we are manufacturing the product depends on the consumer requirement. Hello. Hello. Hello.
Yes, sir.
Yeah. Hello.
Yes,
You
sir.
can hear me?
Yes,
Okay.
sir. You're audible.
Anytime you have cut down, please inform me.
Yes.
So
Sir,
I can.
we can
Hello.
maximize the screen.
You can maximize speed.
No, screen. The screen which you is sharing,
Huh.
is it able to maximize? Because it
I'm
is
not.
the present. Please on the presentation mode.
Yeah. Now.
It is visible, sir. The screen is
Now it's
visible.
visible. So you can see some. You can see on this slide this small boy which is trying to wear the shoes basically, which is not fitting to the him. So the idea is that. When you purchase apparels basically, okay, you will go to the shop and the first thing what you see that the fitment basically. So if I want to go, I will ask the 40 number shirt basically, or shoes a particular number, but unfortunately that approach is absent in the consumers when you will purchase the personal care product like shampoo, lotion, or any other, I could say, okay, oil. These product we will never have think in that way. Sometimes we'll purchase this product, we'll see, if we try it and say that this is not suitable for my skin or hair, then we will stop that basically. But while choosing, hardly there will be 5% consumers who has aware about their skin type, hair type, scalp type, and their skin issues. Now why? Because lack of awareness, lack of hand holding basically. There's no hand holding for the consumer. That the reason consumer doesn't understand what is his actual requirement. So he'll purchase the product, personal care product, he will try, and at the end he will come to the conclusion that this product is suitable to me, his product is not suitable. So what we can do here? So I hope I will not give the definition of cosmeceuticals. I will go to the personalization and customization. So what is difference between personalization and customization? Personalization is nothing but where product has been designed on the basis of individual need. So here your skin, your hair type will be get analyzed, your body will get analyzed, and formulation has been done. This is customized basically. This is personalized. Customized is nothing but we have a largely we understand that what are the different type of the skins like dry, normal, oily, like that, or sensitive, and we will design the product. So whole population will be divided into 5 or 6 types, and basically from that you will choose the product. Is called as a basically customization. Now how this total market of personal care products. So personal care products are nothing but so I'll make it very clear that what is personal product basically, and what is a cosmetic. Personal care products are those products which you require for the daily need, which will be getting without prescription. We can say And now most of the companies have segregated their product. They believe that created one lotion, the population is not able to satisfied with that, because every individual has a different type of skin and every individual has a different type of skin and every individual has a different type of issues basically. So now we are little customized. We will make a different lotions which will be suitable for different skin. For example, dry skin, for example, for oily skin, maybe sensitive skin, like that we'll make. Or shampoo for very dry scalp, then oily scalp, like that. So now that trend has been started, and before 2031 we assume that out of 15.3 basically, which is growing at this rate, will be surpass this. So whole market will get customized, so nobody will go and directly they'll purchase. Everybody will see that what is their skin type and accordingly they will purchase. So there will be no single product basically. Now what how customized products are good, because they engage the consumers with their own choice basically. It's now addressing skin and hair concerns. Skin type basically, skin type according to normal, dry, oily, we can get customized our product. Hair type, what different type of hair, wavy hair, straight hair, curly hairs. Scalp, which is dry, very dry, extreme dry, dandruff, so by which we can divide. So when you design the product, basically you can design the product based on the consumer's type. So I hope everybody is understanding this. So when you design any personal care product, you will see that the product will be suitable for different people. You not develop the one product which will be going for everybody, because you know that everybody may not be able to fit or give us 100% satisfaction or close satisfaction to their expectation. Now first I just show you that how the product can be categorized. Now what are health concern basically? For example, sensitive, allergies, pregnancy, these are some concerns. Lifestyle, some people have resist sweats always, so they want some product which resists the sweat. Or environmental exposure. Personal preferences, for example, fragrances. So if you go to, I'm just because I'm working with this organization, so in our product basically, when you go to shampoo, you will see that in shampoo you will ask them that what is your fragrance requirement, which fragrance you like. So we have around 4, 5, 6 fragrances. From this, consumer will choose the fragrance, consumer will choose the color, and they will go ahead in that way. So what we'll do, we will get the shampoo base. For example, somebody has oily skin or oily scalp, they will get the shampoo which is suitable for oily scalp. I'm not going to detail in the formulation, because oily scalp requires lower percentage of a surfactant, because drying issue will come. So that basically will add this fragrances and color choice basically of the fragrance of the consumers, and will give. So it's more or personalized kind of. So that is required. Now adapting product which local climate. So now at one point, if you see the climate of Konkan, for example, Maharashtra, if you see a Konkan, the climate is different. Mumbai climate is different. The Latur climate is different. Maybe South Maharashtra, basically, the climate is different. So for that, you need to design the product according to the target climate. So one product which manufactured at one place will not be suitable if you go to other place, which is climatically different. UV exposure, for example, you are going to Goa, for example, where when you go there, most of the product you can see are SPF basically, which is sun protection factor. So it's more or role around your exposure to the sun and protection. Water quality, where we have very hard water will be there. TDS will be very hard. So you need to design the product. Suppose your target is there, or you're targeting product basically to the particular region, you need to understand the water quality. So what I want to say, that these things has to consider when we design the product. So one product will not able to fit to the all consumers. Now season wise, if I come down to the season wise, winter, summer, and seasonal adjustment basically. So one product which give me a satisfaction at one season, for example winter, maybe summer I will not like that product, because of the weather, because of the environment, seasonal changes, the product is not giving the same kind of benefit or satisfaction, I could say, or objective towards the objective. So these things are there, where humidity will increase, skin texture will change, maybe sun intensity will change. So this has to consider. So at one point when you design the product, so there are some so there are some companies, India we don't have that trend, we started just now, we are working on this, where suppose one brand you have, okay, so one brand, for example, you have sunscreen. So this sunscreen will be satisfied, there will be a formulation will be different at a different time. So every 3 months, the formulation will change because seasonal requirement has been changed. So one formula which fits now, maybe not necessary that it will be fitted to the next 3 months, because for the same person, maybe today I will say this is very good, but maybe possible that afterwards I'll not. It's like some foods, for example, if you eat at particular season only, you like it, because in other season maybe your body requirement changes, body has been asked something, demand is different. So same thing is applicable for your personal care products also. So we have provision that every 3 months the product composition will be changed to suit the environment. Now, most important, which will be very close to today's topic. Yeah. Hello, Professor Pawar. Hello. 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 Hello.
Hello. Yes, sir. Yes, sir.
Yeah, I'm going in the way. You receiving the things?
Sir, yes, sir. But the slides, they are not moving, sir. That is, the slides are not sharing. Means we have to click on the slides. Yes, yes, yes.
Uh, I'm not now. Now you can see.
Or you can click on F5, sir.
Now you
The
can see,
next
sir.
slide is visible now, sir.
Now you can
Yes, sir.
see.
Yes, sir.
So, yeah.
Yes.
Yeah, so those who want to ask a question in between, they can ask. I'm very happy to interact basically. So I don't want a monologue basically where I will speak and everybody will listen. So if you have any question at any point you want to ask, you can please ask me. My session is not very scientific basically, moreover practical approach are there, and I just add the AI leg in my work. So what my organization is doing, what I am doing, I just add that how AI is helping us in a day-to-day life. So it's not very theoretical. So you can ask me a question, I'll be happy to answer you, and that by which I understand that whether you are getting this or not, like that. So I'll just recall the earlier. So what we have done, we have seen that how skin basically different, hair are different, scalp different. Then we said that because of lifestyle, their needs are different. Then I just show that because of the different climatic conditions, UV exposure, water quality at different places is different. So that's the reason you need to design product accordingly. Then you have a season basically. So one product is not able to satisfy at the all seasons. So maybe your product need to change as the same as a food basically. So one food, for example, you feel that in summer you feel to eat ice cream basically, but if you give that on winter, maybe you will not happy or you will not take it. The same thing is applicable for the product. Why not applicable? The question should come in mind that why not applicable to my personal care products? They should have changed the composition, they should have changed the formulation according to the seasonal needs. So this principle we apply here. Now coming down to the AI, like very important, that how this is helping in personal care products. So you can see here that so one screen is there where the face has been shown. So by which what AI will helping. Now if I ask the lame person basically, or maybe educated person, that what is your skin type, people, 90% people get confused how to identify my skin type. Sometimes I feel that my skin is dry, my skin is oily, my skin is normal. So this is very difficult because of awareness, because of lack of understanding that how to measure this basically, and what are the parameter to measure the skin type of individual basically. So AI will help you to understand. So generally what we'll do, we will have a system where consumer will get this face basically exposed to this camera, and where they will analyze that what is your skin type basically, what are the skin issues, even how the basically the lines are there, the wrinkles are there, the depth of wrinkle basically, how many pimples you have, where I could say the anti-aging product basically, we can say underline product, we can say under eye product basically, where dark spots are that. So they will check that whether it is normal or it is very high, that kind of thing. So by which all these, they will understand that what is your requirement. So AI will read this by different algorithm. What is mean by algorithm basically? Now coming down to question, algorithm basically will ask some questions to the person. Now this will be applicable for Ayurveda as well, where ask the what is your sweat level, what is your appetite, basically what is your hair volume, what type of your hair, ball patch is there or not, then basically bowel movement, then what, how you use network, you are alone person, that kind of questions will ask, and they will understand about your psychology as well by AI. And back end they will understand that what is your requirement, what kind of formulation you need basically. So again, I'm if I'm telling you, for example, I'll give, because we are come from pharma, so that's the reason I will give the direct example. For example, somebody has a very dry skin and somebody has very oily skin. Are you giving the same lotion basically to the same both person? Is it what, or otherwise what you need to do with this formulation? Can anybody tell me? Hello, 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 hello.
Hello, sir.
Yeah, so
Hello.
I have asked one question.
Yes, sir.
Hello.
Hello. Yes, sir.
Yeah. So I asked one question. So I was asking that somebody has oily skin, somebody has a dry skin. So does the one lotion will give you satisfaction? Of course not. So what changes we required in the formulations to give you satisfaction, and what are the formulation changes required in both basically? If you want to develop a two lotions, one for dry skin, one for oily skin, so what are the formulation changes required in both? Can anybody answer from you?
Is the question for all of the participants involved.
Yeah,
If anyone
yeah,
is having the answer of that question, which Dr. Husli sir has asked, can raise
yeah,
your hand.
I,
We will
yeah,
unmute you from our side.
yeah. I think Dr. Savita has answered. Yeah.
Yes.
So basically, yeah, to some extent, let me. Hello.
Hello, sir.
Yeah, yeah.
I think so. There was
So
some technical issue.
yeah. So I think somebody has answered. Yeah. So basically, when you have a dry, when you make a formulation which is for a dry skin, your TFM basically total fatty material in the lotion will be on the higher side because you need a more moisturization basically from that. So I think the answer is the in the right way. When you are developing the product lotion basically for oily person, you don't have need a moisturizer basically in the higher concentration. So your TFM will be very less basically in this condition. But for a dry, you need some moisturizers which will be long lasting. Whereas the lotion which we make for oily, you need something which will able to control your sweat production basically. So these things has to consider basically. Now, AI will help you to get the online tools will be able to understand that what is your requirement. They will also help to create a personalized formula. For example, you have acne. So may not necessary that acne has a different one stage. It will be a different stage, or it may be extreme stages. There are different type. I will not go in a deep detail, but AI will tell you that what kind of acne has person has, at what stage it will be. So accordingly, the dosages of the active concentration will be adjusted and will give. So AI will help you to make the product which will be close to the requirement of the person. Okay. Yeah, Professor Pawar. Hello. Hello.
Yes, sir. Yes, sir. Yes,
Yeah.
sir. You're audible, sir. Yes.
Yeah, you can see the screen.
Yes, sir. It's visible.
Now, yeah, AI driven feedback system basically, how AI is helping, in which for example, AI will able to help you to find out this is happening in current scenario. There are some brands, including our brands, which is doing some of the things here. AI will able to understand what is your predictive analysis called as what is your future requirement. So suppose the age of 20 person want person's skin and hair, or basically other health has specific requirement over the 5 years or over the 10 years. Your requirements are changing over the period because the body will be doing some is the changes. So accordingly the skin and hair and part other things change. So from your other parameter that what is your requirement. So maybe one person is taking product now, so maybe it's happened that after 5 years the AI will able to tell you that